This protocol was originally published by Dr. Gary Bunker and colleagues, and allowed for the low density and high purity culture of embryonic rodent hippocampal neurons by suspending the neurons over a glial feeder layer. This method is advantageous over other hippocampal culture methods because it enables neurons to be grown at low densities with few contaminating glial cells. This method is therefore ideal for high-resolution imaging and functional assay of developing and mature neurons in culture. Visual demonstration of this protocol should expedite its adoption by new labs because there are several key steps during the procedure that are difficult to describe through text alone. This protocol consists of 3 independent populations that are required for culturing high-quality primary neurons. These processes include glial cell culture, cleaning and coating of coverslips, and the preparation of hippocampal nerve cells.
To begin, early postnatal rat cortical tissue should be obtained as described in the written protocol. While not directly shown here, note that the dissection steps shown later in this protocol are nearly identical to those used for the cortical dissection. For this reason, relevant steps will be highlighted during the hippocampal dissection procedure shown later in this video. Beginning from step 6 of the written protocol, remove the HBSS from the collected cortical tissue and then mince the tissue as finely as possible using scissors. Add enough